DNA Depot presents Kit DD402, Diffusion and Osmosis. In this experiment, students will learn how molecules selectively move by the process of diffusion and osmosis. Semi-permeable dialysis tubing will be used to demonstrate the movement of different molecules within and through cells. The contents of this kit include dialysis tubing, green dye, orange dye, calibrated transfer pipettes, and salt packets. You will also need a 250 milliliter beaker, jar, or disposable clear plastic cups, distilled water. Grocery store distilled water is acceptable, scissors, and a ruler. Pre-lab preparation. 1. Cut the dialysis tubing into 5-inch segments. You should obtain 18 segments for 6 sets of 3 experiments. 2. Soak the segments of dialysis tubing in a clean beaker or jar filled with distilled water. Soak the tubing for at least 1 hour or overnight. Activity 1. Learning about osmosis and dialysis membranes. 1. Carefully tie an overhand knot at one end of a segment of moist dialysis tubing. Tie the knot approximately 1 inch from the end. Repeat the same for a second piece. 2. Prepare 2 water baths by filling 2 clean 250 milliliter beakers or disposable clear water cups or jars with distilled water. 3. Fill the first dialysis bag with 1 milliliter mixture of the green dye as follows. Roll the untied end of the dialysis bag between your thumb and pointer finger to open the end of the bag. Squeeze the calibrated transfer pipette bulb to fill it with dye to 1 milliliter. Gently insert the tip of the transfer pipette deep into the bag and gently transfer the dye solution in the dialysis bag by applying pressure on the bulb. Remove trapped air in the dialysis tube without displacing the green dye. Carefully tie the second knot at the open end of the bag. And gently place it in one of the water baths. 4. Rinse the transfer pipette with water in a separate container. Do not use the water baths in which the dialysis tubing will be placed. By repeatedly squeezing the bulb and flushing the pipette until totally free of the dye. 5. Fill the second dialysis bag with the orange dye. Follow the same procedures as outlined in step 3. You should now have 2 filled dialysis bags, 1 green and 1 orange. Place each bag in a separate water bath. 6. After 15 minutes, observe if there is any color change in either dialysis bath water. 7. Subsequently remove each dialysis bag and observe its contents. Is there any change in the color in the water bath and/or the dialysis bag? Put the dialysis bag back into the water bath. 8. Periodically, over the next 3 hours, check the color of the dialysis bags and the water baths and determine if color changes occurred. Save the dialysis bags that contain the 2 dyes in their respective baths until you are ready to perform Activity 2. Record your results. Conclusions. Now that you have learned how semi-permeable membranes allow different size molecules to pass, you can see how this is applied to how cells bring in nutrients or remove waste. We hope you have enjoyed DNA Depot Experiment 402 and learning about diffusion and osmosis.